Hello, and welcome to ACR Journals on Air podcast. I am your host, Vicky Shamagam. The old saying, trust your gut, is typically used to refer to intuition, but few of us think about the nerves in our gut, until that is, something goes wrong. The manuscript we will discuss today is Anti-Gefrin Antibodies, a novel specificity in patients with systemic sclerosis and lower bowel dysfunction. It was published in Arthritis and Rheumatology, and a link to the manuscript and disclosures are in our show notes from today. I'm excited to be joined by the manuscript's first author, Dr. Zsuzsanna McMahon. Dr. McMahon is an associate professor of medicine at the University of Texas Health Science Center in Houston. She's a graduate of UT Houston and completed internal medicine training at the UT Southwestern Medical Center in Dallas. She then completed her rheumatology fellowship at Johns Hopkins School of Medicine and a master's in health science at the Hopkins Bloomberg School of Public Health. She was on faculty at Hopkins for about 11 years before moving back to Houston in 2023 to become co-director of the UT Houston Scleroderma Program. Dr. McMahon co-leads the gastrointestinal working group of the Scleroderma Clinical Trials Consortium and also co-leads the gastrointestinal ad hoc committee of the World Scleroderma Foundation. She has been very actively involved in the American College of Rheumatology, having previously served on the board of directors, and she currently serves as the chair of the ACR Committee on Finance. Welcome to the show, Zsuzsanna.
Thank you for having me, Vicky. I'm delighted to be here.
So I really enjoyed reading your manuscript for two reasons. Obviously, first and foremost, I always love learning new things about rheumatology, and who doesn't love a new antibody? But I also wanted to give you a shout out for your article's lovely linguistics. It's a very enjoyable read. In the introduction, you posed some intriguing questions. Tell us what they were.
Oh, I appreciate your kind words, Vicky. Thank you very much. Well, the gastrointestinal tract is one of the most involved organs internally in scleroderma. And as you know, patients present with a variety of complications from mild heartburn to recurrent lower gut pseudoobstruction. And normal intestinal motility is controlled by neurons and glial cells of the enteric nervous system in association with other cell types in the gut. And what's interesting is that prior studies have shown that antibodies to muscarinic 3 receptors in scleroderma, which are expressed on smooth muscles in the gut and nerves, associate with rapidly progressive GI disease and pseudoobstruction. And prior studies have also shown in scleroderma that patients without extractable nuclear antigen antibodies, or ENAs, had a higher scleroderma GI symptom burden, suggesting that antibodies that are yet to be discovered may be associated with scleroderma GI severity. And so we were really interested in identifying previously undefined antibodies in scleroderma-related GI disease to determine if there was a correlation between scleroderma antibodies and gut function, as this would potentially suggest that scleroderma antibodies target autoantigens expressed by the ENS and associated GI cells.
And so your story begins with a single patient. Did you have a sixth sense that this patient was going to lead to an amazing discovery, or were you just trusting your gut?
I love that. We did, in fact, have a gut feeling about this patient. But in all seriousness, this was really just the perfect patient. So it was the only patient with severely delayed lower bowel transit in this cohort of patients with whole gut transit studies that we had that also had a completely negative autoantibody panel. And so we found this patient really interesting. Briefly, we weeded out the other patients as there were 20 others with severe GI delays. 10 did not have complete antibody panels available to us, and so we were less interested in exploring their SARA. And among the remaining 10 patients, 9 had identifiable scleroderma autoantibodies. And because we were really looking to perform antibody discovery on a patient with severe GI disease and no known specificities, this was the ideal candidate for our discovery pipeline.
Yeah, makes sense. And then, like all good tales, you also have a mouse. This one is called Tomato, or should I say tomato? Tell us about the mouse. What made it unique? And how was the mouse so important in this study?
Great questions. And yes, we must credit this little mouse. To answer your question, the mouse is important because the Wnt-Cree TD tomato lineage fate mapping mouse model labels all neural crest-derived derivatives in the gastrointestinal tract with a red fluorescent reporter TD tomato label. So, enteric neurons that help control GI motility are derived from the neural crest cell lineage. And this mouse was important because it's challenging to access the deeper neuromuscular tissue in the human gut because of risk of GI perforations and such associated with these procedures. And in this case, the mouse shared its longitudinal muscle and myenteric plexus layer with us and allowed us to conduct our study.
So let's jump now to the discovery phase of your study. Can you tell us how you identified the enteric nervous system autoantigens and how you use those antigens to then identify antibodies in your patient?
Great question. So we used lysate from this longitudinal muscle and myenteric plexus layer of the mouse we just described, and used the aforementioned serum from the patient who was EN8 negative and had severe GI dysmotility as a probe. We then utilized immunoprecipitation on bead digestion and mass spectrometry to identify the autoantibody.
And so then you looked at a cohort of scleroderma patients and some controls to look for anti-Gefrin antibodies in a larger cohort. Can you tell us about those various cohorts that you studied in this part of the work?
Sure. So the scleroderma all-comers cohort consisted of all comers, essentially consecutive patients with scleroderma, regardless of their GI status, who were recruited during routine clinical visits from April 2016 to August 2017. And that was about 188 patients. This allowed us to examine the prevalence of the antibody in scleroderma and look for associated clinical characteristics. We also looked at sera from 40 healthy controls to determine its prevalence in the non-scleroderma population. And then to evaluate whether the antibody was more specific for patients with scleroderma compared to others, we also looked at the prevalence of the anti-Gefrin antibodies in a cohort of patients without scleroderma, but with GI dysmotility, and a cohort of patients with necrotizing myopathy. Finally, we examined the prevalence of this antibody among patients with the most clinically severe GI dysmotility, or those on TPN or parenteral nutrition, to see if it was enriched among patients with more severe GI disease as well. We ultimately found it was highest among these cohorts in the scleroderma all-comers cohort.
So let's talk now about antibody titer, because I think you evaluated the relationship between antibody titer and symptom severity. And the patient outcome tool that you used for this component of the study was the UCLA Gastrointestinal Questionnaire, which, for those who don't know, is a lot of questions about the bowels. Tell us about that questionnaire and what did you find?
So the UCLA GIT 2.0 is a validated patient-reported outcome measure assessing GI symptoms and their severity in patients with scleroderma. In our study, we found that patients with anti-Gefrin antibodies were more likely to have significant, meaning moderate to severe constipation, compared to patients without this antibody.
Okay, so let's get back to the mouse. You did some immunostaining in both the mouse and the human samples, and I urge our listeners to go look at the Technicolor in the manuscript because a picture is worth 1,000 words. But Sujana, can you describe for our listeners what you show in Figure 2?
Sure. So first, we wanted to visualize the cellular targets of anti-Gefrin antibodies in the gastrointestinal tract. So we used sera from patients with scleroderma who were anti-Gefrin antibody positive to immunostain the longitudinal muscle and myenteric plexus tissue sections from the mouse. In this system, we had the neural crest-derived cells, again labeled red with the TD tomato. This clearly labeled the enteric nervous system. And we also co-stained with commercial anti-Gefrin antibodies, confirming that the patterns observed with patient sera were consistent with Gefrin expression, both labeled enteric neurons in this layer of the gut, as well as endothelial cells in the murine GI tract. And then to address whether our observation of Gefrin expression by murine enteric neurons extended to human tissue, we performed immunohistochemistry on paraffin sections obtained from distinct human GI tract regions, and Gefrin was detected in the longitudinal muscle and myenteric plexus layer in a very similar distribution to the muscarinic 3 receptor, which is also expressed by enteric neurons. So we were able to validate our findings and show that both were expressed in enteric neurons.
That's great. So tell us what this means for clinicians. What's next for getting anti-Gefrin antibodies from the lab to the clinic?
Well, so it's not quite ready for prime time yet, but it's tantalizing to consider the potential value that anti-Gefrin antibodies may ultimately have in clinical practice. So if anti-Gefrin antibodies are fully validated as biomarkers and are demonstrated to be consistently present prior to the onset of significant lower GI disease, they could be used as tools to identify patients who are at high risk of lower GI disease progression. If demonstrated to impact motility negatively, they could potentially serve as markers of scleroderma GI disease activity and targets of novel therapy. But further studies evaluating the role and function of these antibodies in scleroderma GI pathogenesis are still needed before they enter the realm of clinical care of patients.
So exciting. You're kind of giving us a cliffhanger, waiting for more, maybe diagnostics, maybe treatment, it sounds like.
Yeah.
So I want to move this discussion in a slightly different direction, maybe get away from the bowels for a little bit and talk a little bit about your career. Firstly, I wondered if you could share with our listeners why you became a rheumatologist and what drew you to scleroderma.
Great questions, Vicki. So I became a rheumatologist because of the mentors and the patients I saw along the way. I remember rounding with Dr. Frank Arnett as a medical student when I saw my first case of scleroderma. And I really admired his bedside manner, vast knowledge base, and the questions he raised about the biological mechanisms that underlie this really interesting disease. And this was really the first seed that was planted. And as I went through my training at UT Southwestern, I considered other specialties, but realized that the cases that interested me most in these specialties were the ones that were tied to rheumatology. So it was at that point that I chose this subspecialty. And then ultimately, I was drawn to scleroderma when I was at Johns Hopkins working with Dr. Frederick Wigley. And we shared a patient who was a young man with very severe aggressive scleroderma. And no matter what we tried, the patient's disease progressed. He ultimately died, and it left me feeling heartbroken and frustrated. And I remember speaking with Dr. Wigley, and he said, well, Zsuzsanna, you can either do something that is a little less emotionally straining, or you can try to do something about it. And so that challenged me to do something about it. And here I am today.
Yeah, so a mentor to many, Fred Wigley, but he really threw down the challenge to you. That's amazing to hear.
Yes, he did.
Early in your career, you also became quite active in the American College of Rheumatology as a volunteer. Can you tell us about that? How did you get involved? And do you have any tips for other early career rheumatologists who want to volunteer?
Sure. So my involvement with ACR began when I was applying for the fellows subcommittee. In August of my first year of fellowship, I received the email and it really interested me. And I asked my program director, who was Alan Gelber at the time, for a letter of support. And that was when it all started. At the time, the fellow subcommittee members were also assigned to a standing committee, and I was assigned to the annual meeting planning committee. And I believe you were on it at the same time as I was. And that was probably when we first met, actually. And the committee was so supportive of new ideas, even from fellows. And so I proposed the ACR Knowledge Bowl at the time, and it took off. And I'm so thankful for the time I spent with ACR over the last 15 years, because it's been truly life-changing and special in so many ways. And I've met people like you and others throughout the college who have really inspired my career, become great friends and mentors. And so I highly encourage other early career rheumatologists to become involved, because it's fun and a great opportunity to contribute and grow and meet people outside of your institution.
Oh, I think that's so true. And you're right. We did meet through the fellows subcommittee. And I would urge all fellows to consider applying for that subcommittee. It's great fun. You will get kind of distributed across different committees of the ACR. You get to see how the ACR runs, which is quite eye-opening for people at the fellowship level. You're right that both you and I had the good fortune to serve on the annual meeting planning committee, which is probably the most fun committee, I would
Yes, I
say.
agree.
I'm biased,
Yeah.
but there's sort of great opportunities there, and definitely urge our fellows to apply. From that role as a fellow subcommittee member, you then became a member of the board of directors of the ACR. Tell us about that experience. How did you get nominated for that role? Because I think you did it straight after fellowship. What was it like, and what did you learn?
Thanks for these questions, Vicki. I really enjoyed my time on the ACR board of directors. I was quote unquote young board member at the time. And I'm not exactly sure how I was nominated for the role, but it was a wonderful surprise to get the phone call. And my task at the time was to represent younger members of the ACR and provide relevant perspectives to the board. And it was a wonderful learning experience for me to see and interact with such leaders in our field and learn from their perspectives and learn how to navigate some of the complex topics that were raised in board discussions. And I remember being inspired at the time to see firsthand how much the ACR senior leadership, both physicians and staff, really cared about young and incoming rheumatologists and tried to find as many ways as possible to support their growth in the field. And I remember I was a young investigator at the time, which was a pretty daunting time in the career. Anything can happen. And I truly felt like the college had my best interest at heart and was doing everything in its power to make life easier for me and others on this track, as well as for the rising clinical rheumatologists. And so they truly have a heart for the next generation. And that was when it was most apparent to me how unique this organization truly is.
Oh, I think that's so true. I'm going to jump forward in your career now because you've also successfully navigated what's fondly referred to in the business as the valley of death. And by that, I mean you have successfully transitioned from junior investigator to independent investigator. And with that, you recently moved institution, which is also an extremely complex process. How did your intuition help you along the way? Was it all balloons and rainbows, or were there any bumps?
I've never heard it called the Valley of Death before, but I can see where that would be an appropriate term. So, the intuition is actually an important part in the process. It obviously takes more than intuition, but somehow the timing has to feel right. And I will say the right opportunity came at the right time. And the UT Houston group, the NIH team, and my colleagues at Hopkins are actually all very helpful in navigating this process. You're right that it's quite complex and required a lot of work to get the grant transferred and up and running. In fact, that just now happened the last few weeks. But Hopkins kindly released the grant, and UT Health had the infrastructure in place to support me throughout the process. And I was able to hire a research coordinator who was an incredible help throughout this effort. There was a lot of stress throughout, mainly because I had been at Hopkins for 15 years and I was saying goodbye to many friends and moving to a new city. But I was excited about my next steps in the R01 and next steps in the career. And I think my colleagues on both sides made it incredibly helpful. I also had family in Houston, which was very welcoming and made the transition a bit easier. So I feel very blessed in that regard.
You really were going home, I guess.
Exactly.
And do you have any tips for listeners who might be considering moving institutions? Any kind of good advice for people navigating that?
Sure. So it can be a tough process to navigate balancing your existing relationships where you currently work and then new relationships in the place where you're about to work. And I really relied on my network of rheumatologists, many of whom I've known through the ACR. And they helped to guide me through the process. I also spoke with former colleagues and family members who are in the medical field and were also quite helpful to me. So I really pulled on the knowledge base and experience from a lot of brains around me. And I was fortunate in having a supportive group of colleagues at Hopkins who helped me in the time of transition, making it feasible for me to wrap up my research projects there as well. So I'm very thankful for that.
And then obviously kind of transition of patients is always complex too, right?
Exactly. Yeah, no, I was really sad to say goodbye to my patients there, but many of them have come with me at least to check in periodically, and I still stay in touch with many of them. So that's been a really nice part of the transition.
Well, Sujana, I wanted to thank you for sharing your work and your story with us today. Chatting with you has been an absolute pleasure, and I'm really looking forward to hearing more about your work in the future. I guess your R01 has just started, so we have another 5 years of scleroderma antibodies to come.
Thank you so much, Vicki, for inviting me to join you today. I really enjoyed being part of your podcast and always enjoy listening.
Our thanks today also go to the many staff and volunteers who make the American College of Rheumatology a great organization. The work of the college can only happen with the whole team's hard work. And the excellent news for rheumatologists who volunteer is that you get to interact with ACR staff, who are, hands down, some of the nicest people I know. Thank you all for listening to this podcast. ACR Journals on Air is a production of the American College of Rheumatology. Please follow at ACRheum on social media, email us at podcast@rheumatology.org, and rate and subscribe to our podcast at acrjournalsonair.org or wherever you get your podcasts.